We're now going to talk about the process of B cell selection. I made a separate video for T cell development where I talk about selection a little bit, but I want you to keep in mind that the concepts are the same. This is really referred to as tolerance. Okay, so anytime we're talking about whether or not a T cell or a B cell was selected, we're talking about tolerance. And when I talked about T cells, or when I talk about T cells, if you haven't watched that video yet, that's okay. You didn't miss anything. Keep reading. It's the process of central tolerance. And central tolerance is really just determining, is the antigen receptor we made a safe antigen receptor? So this, I think, is very similar to kind of your own journey to medical school, right? You were developed, you went to undergrad, you probably did some shadowing, and then you were tested. You took the MCAT, you went through a rigorous interview process, and then you were selected. We selected you. We invited you to come to our medical college, and that's the same process really for lymphocytes. We develop them, and now we're going to test them before we kind of cut them loose and see what they can do in the body. Okay. For T cells, the process is known as positive and negative selection. And those are processes of central tolerance. It's referred to as central tolerance because it takes place in the central or primary lymphoid organs. So for T cells, that's going to be the thymus. And for B cells, this process is going to take place in the bone marrow, right? Because that's where those cells develop, and we're going to check them there, and that's central tolerance. Okay, so remember, when we were creating these antigens, our first goal was to generate a diverse antigen receptor. Then we needed to alter or eliminate any self-reactive B cells or B cell receptors, and that's what we're going to do now. And then at the end, I'm going to talk a little bit about promoting that foreign reactive B cell. So just like you do at the end of your medical degree, the B cells and T cells that we develop take kind of a Hippocratic oath. They choose to say, first, do no harm. I was actually really disappointed when I first heard the Hippocratic oath that apparently you guys don't even say that. But I like it better, so I kept it in here. So do no harm. That's your first goal as a lymphocyte. Apparently not this way. Okay, so how do we do this? We need to figure out what the self-reactive or autoreactive cells are. So these are cells and receptors that react against a person's own tissue, be it pancreatic islet cells, or myelin cells in the brain, cells of the optic nerve, whatever it is throughout the body, we need to make sure that if this B cell comes across it and it's self and it's safe, that we're not going to attack it. And self-antigens are basically the antigens that these receptors might react to. So we need to shut down this process altogether. All right, so this is where the problems come in. We used a completely random method for creating antibodies or antigen receptors made out of material very like the material we're trying to protect against. So now we're going to get into tolerance. And this is the mechanism that will occur in the bone marrow so that we do not produce cells that will attack our own tissue. Okay, so how do we do this? If an immature self-reactive cell, so these are immature B cells in the bone marrow, if it was released from the bone marrow and set loose upon the body, activation and maturation of the B cell would produce self-reactive and harmful antibodies that could cause disease, and that's termed autoimmunity. To prevent this from happening, B cell receptors of immature B cells are wired to generate negative intracellular signals when they bind to antigen. So first off, it doesn't get released because it doesn't get cued to undergo class switching to produce IgD until after it's done this testing. So IgD is basically the B cell's license to drive. This is what gets it out of the bone marrow. So remember, I told you initially that for a B cell, initially it's going to express IgM on its surface. After it's been tested and gone through its selection process, it's going to co-express IgM and IgD. And once it has IgM and IgD co-expressed on its surface, using a process known as alternative splicing. That's how it's able to express both. Once it's done that, that's when it's actually released from the bone marrow. If the cell is not self-reactive, if during the testing process it is found not to be self-reactive, then you have alternative mRNA splicing of the heavy chain gene transcripts to produce the IgD on the surface. And it's this dual expression that leaves. So you can see that an immature B cell is going to move to the blood when it expresses IgM and IgD. If it only has IgM because it's reactive with a self-antigen, it's going to be retained in the bone marrow and eventually die. So what is the testing process like? It's really about strength, okay? If the IgM on the surface binds to a self-antigen that is present in the bone marrow strongly to a single self-antigen, and it's going to be abundantly expressed throughout the body and therefore also present in your bone marrow through the work of AIR, that autoimmune regulatory expression gene, then we're going to see death. This is known as clonal deletion. So basically any cell that responds really, really strongly to a self-antigen is going to be killed, and that's clonal deletion. Now, here's where things get a little bit sneaky, okay? If the cell binds kind of moderately to a self-antigen, right? It's going to undergo receptor editing. Now, I talk about this when I talk about making the antigen receptors, okay? So it sees the self-antigen and it binds it moderately. So what it does is it goes back in and it tries to rearrange the light chain genes. So literally, if it was V1J2, it's going to go back and say, okay, that combination led to a moderate strength response. So let's try something new. Now let's do, say, V2J4. And it'll go and it'll test it again. If it binds strongly this time, it'll die. If it binds moderately, it'll try again. It'll say, okay, we still didn't get it right. Let's try V3, J5, and so on and so forth until it uses up all of the kappa light chain options and all of the lambda light chain options. If it runs out of genetic material, the cell dies. If at any point it does not bind the self-antigen, then it can go through alternative splicing, get its IgD, and be released into the periphery. And this process, like I said, is known as receptor editing. And it's really, really important, and it's interesting that B cells are able to make these changes later in the process. And this only happens on the light chain. This is light chain only, and that's key to know. And these will be DNA level changes. So once it's decided to abandon V1J2, it can't go back to that ever again. It'll have to go on to the next one and the next one and the next one. All right, so at this point, our B cell did it. It made it through the steps to become a fully fledged B cell. Now it needs to differentiate into a plasma cell capable of secreting antibodies. I liken this to picking a specialty. So one day when you graduate med school, you're going to become like an ER doc or a family practitioner, and then you're never going to go back and do something else. Like that's kind of what you are. We're going to go into this in a lot more detail later, but I thought I'd kind of outline the 3 basic steps that are going to take place for our little B cell now that it's made it out to the periphery. So the first thing is it's going to meet its antigen. So this is true love. It's the right antigen for the right receptor. Now, these aren't T cells, so this is going to happen directly, right? We don't need MHC in this interaction, no cross-linking, nothing. So we're going to see cross-linking of the antigen receptors. and that's going to lead to activation of the B cell. If there are some helper T cells around, they're going to contribute cytokines. And those cytokines are basically the marching orders. They're going to tell the activated B cell what it should basically become. The activated B cell will proliferate, and based on the messages it got from the cytokine, it will differentiate. If the T cell was present to give a differentiating signal, it will class switch into a plasma cell capable of secreting antibodies other than IgM. So IgG, IgA, and IgE. Those are pretty much your main ones. If T cells were not present and no cytokines were given, it can only ever produce IgM. We'll talk about this in a lot greater detail, but that's kind of the plan for our B cell now that it's graduated.